Okay, so we're here today at the 86th Cold Spring Harbor Symposium on Biological Timekeeping, which is being held virtually this year. So my name is Luke Smith, and I'm an associate editor at PLOS Biology. And today it's my great pleasure to interview Dr. Achim Kramer, who's actually a member of our editorial board at PLOS Biology and who also serves as an editor at PLOS Genetics. Dr. Kramer is a professor at Charité University, where his lab has helped tease apart the molecular mechanisms governing the circadian clock in mammals, as well as their impact on physiology and behavior. So today I'd love to hear about some of your work on this topic and some of the interesting tools that you've been developing to aid with this research. But first, I was wondering if you could just tell us briefly a bit of your background and how you became interested in circadian rhythms.
Yeah, thanks, Luke. It's a pleasure to be here. Actually, I did a PhD in a completely different field. I was working on peptide libraries and on structural biology, and I did even a little postdoc in this subject. And then that was the end of the last century. Everyone said, I mean, the next century is the century of the brain, and I was looking for kind of switching fields, and I wasn't sure what to do. And I read in a popular newspaper in Berlin about the discovery of a new clock gene. And at the time, in the 90s, that was a paper from Chuck Weitz together with Claude Takahashi. They discovered B-mail with a yeast-2 hybrid screen. And I was puzzled and stunned and thought I'd never heard about a clock. And I emailed Chuck and asked him for an interview. And that's how I came into the field. So my first clock experience was with Chuck Weitz at Harvard Medical School, where I stayed a couple of years, and then I moved back to Charité in Berlin and kind of got an assistant professorship, and now I'm full professor for chronobiology there.
And do you bring your background from your PhD into your current work, or are you pretty far from that now?
Well, I'm always interested in structural biology. We have been collaborating with Eva Wolf a couple of years ago, where we published the crystal structure of a PER2-Cry1 interaction in cell. At the time, that was very what was kind of a flashback into my old field of looking at structures and amino acid side chains and hydrogen bonds and salt bridges and so on.
Nice, nice. So at this conference, you presented on a very interesting project, which is developing a new approach for the characterization of endogenous circadian clock protein dynamics. And then you can do this in living cells, which I think is really interesting. And so I was wondering if you could tell us, a bit about this and what you found with this approach. But first, maybe you could explain why you set out to develop this system and kind of what kind of questions you've been asking with it.
Yeah, our motivation was we realized that the cell biology of circadian clocks, with that I mean, where is the clock protein in the cell, at which time, and when does it interact with whom? This was really not really developed. We have been done in the past some efforts to kind of tag clock proteins with fluorescent proteins and then overexpress it, and then often this destroys the clock. So you can then use the cell only as a container, but you cannot really have a really dynamical view on clock proteins, subcellular localization, behavior, interaction. And when the CRISPR revolution kind of appeared, we immediately thought we should kind of exploit that and make the knock-ins. But that turned out to be difficult. So we tried even earlier with talens and zinc finger nucleases, but this was horribly inefficient. The CRISPR-Cas technology made it now possible, but it was not so easy because these clock proteins, they oscillate, and at some points in the cycle, they're even almost undetectable. So we had to think about a kind of, or combine several tools which were around for our clock proteins to make it work in an efficient way.
So can you describe kind of generally for a broad audience exactly kind of what you've been doing? So you've been tagging these endogenous clock proteins. Is that right, basically?
Yeah. So we to observe these clock proteins and their dynamics over many days in living cells, under the microscope to get the spatial resolution. We tagged them with fluorescent proteins. So, and we did that with green proteins, with red proteins, because later we also want to have them as a so-called FRET pair, where you can use energy resonance to study interactions. So, and we put them. So we made fusion proteins, but the goal was to have the endogenous proteins as a fusion protein. So then these proteins are expressed from their endogenous promoters, which is usually for many of the clock proteins a rhythmic promoter, and then the protein is made as a fusion protein and is not destroying the clock, because if you use a constitutive promoter, which is used in many overexpression studies, then you have high levels of for example, a negative regulator of the clock, like the cryptochrome proteins, and then you destroy the clock and cannot see any rhythms anymore. That's why the knock-in is so important. However, there are also challenges, because in the knock-ins, you never know whether your tag kind of destroys the natural function of the protein. So we had to, for the PER2 protein, there was a precedence from Joe Takahashi's famous PER2 luciferase knock-in mice. I think this is one of the most cited papers in our field, apart from review papers. And many, many labs use it. And we are, as a field, we should be really grateful for Joe providing this mouse to us. And here also the luciferase protein was put to the C-terminus. And also later, also a GFP protein has been tagged to the C-terminus. So we were pretty sure this is not destroying the clock. For the cries, it wasn't so clear, especially because the cry C-terminal tail is known to be really flexible. But we have been looking careful and also saw that these double knock-in, I should say biallelic knock-ins, you can knock in on one allele, you can knock in on two alleles, but if you knock in on two alleles, this does not destroy the clock. So we're pretty sure that also these fusion proteins are functional.
Yeah, awesome. So in your talk, I was really interested to watch the videos of PER and cry oscillate in the nucleus of the cells, and just seeing it in the live cells is really interesting. And I wonder what types of insights have you kind of derived from this kind of refined approach? What have you been learning about how these clock proteins interact and oscillate in live cells?
Yeah, a couple of things were surprising to us, because I mean, in the past, almost everything we knew about the clocks or the clock protein dynamics came from Western blots. I mean, in Western blots, you harvest tissue from liver or from cell cultures, and then you combine 100,000 or 1,000,000 cells, and what you get is the average of a huge population of cells. And what you don't see then is things like how different is the cell to cell in terms of the circadian dynamics. So that the biological noise, you have no idea about that, because you have always an aggregate of many cells. That was a fascinating insight. So how different single cells can be in terms of the phase relation between PERs and cries. So what we found is that PER and cry protein peak a couple hours apart, which has been put forward in the past sometimes also, but it's a little bit inconsistent with the idea that PERs and cries mostly occur in a huge complex, in a huge multi-protein complex, which makes up the negative inhibitor module, which then binds to PER and to clock and B mail and kicks it from the DNA. It was surprising that we found cry almost exclusively in the nucleus, which is a little bit inconsistent with other people's biochemistry insights. But we cannot exclude that there's no cry in the cytosol. There must be some cry in the cytosol, because cry proteins are made in the cytosol, but apparently even at times when PER is not present or undetectable to us at least, then we also see cry in the nucleus, cry1, I should say. This is also puzzling with respect to the idea that the cry proteins need the PERs to go into the nucleus. So it's possible that maybe the other PERs, PER1 and PER3, play a role, and we are making the knock-ins for them right now, and are excited to see whether there's an interesting phase.
Yeah, that's really interesting. Yeah, and so you're able to measure these proteins in the same cell, is that right? You're able to measure cry and PER oscillations simultaneously, is that correct?
Exactly, that's correct. So we tagged the cry with one color, red, and the PER with the other color. So I should say cry1 and PER2, or vice versa. And then therefore now we can, for the first time, I think, visualize both proteins in the same cell, and then we can see we had a really interesting insight, which is not in the paper, but it's that there seems to be an optimal phase relationship. So when we see cry 5 hours later than PER, then the amplitude of the oscillation is especially strong or high. If it's further apart in one or the other direction, amplitudes go down. So that's just a correlation. And we have been teaming up with Hans-Peter Herzl's mathematical group, and he constructed a mathematical model and kind of could recapitulate these interesting insights.
So does that, I mean, does that suggest that there's some kind of bigger cycle going on, like beyond these little cycles? So there's oscillations and then a bigger cycle of oscillations where they have the maximum amplitude, or is that just a crazy question? I don't know if that question makes sense.
It's unclear. I mean, these systems are so difficult to kind of grasp in our linear thinking. So I always say it's a highly nonlinear system, and everything can happen, even chaos. But in this system, I mean, it's too early to say whether this is a design principle or whether this is a coincidence. It's at least interesting. So we have now suddenly tons of quantitative data where the modelers have been begging us for years to make their mathematical models not only toy models, but models where we can really say, look here, these are the exact numbers. We have also done fluorescent correlation spectroscopy where you really can measure the concentration or the number of molecules in the cells, and found that was the second surprise, or maybe the third surprise, that we have like 5 to 10-fold more cry protein than PER protein, cry1, PER2, which is also not compatible. I mean, we knew that it's a little more, but it's the 10-fold more is surprising given the idea that PERs and cries are almost exclusively together in one complex, and the stoichiometry is 1-to-1, which we found also in the crystal structure. So there's a lot of free cry doing things which is consistent with other people's report that cry can, for example, repress nuclear receptors. The cry1 is also acting as a PER independent late repressor for the clock beamer. So all these insights with really quantitative numbers, this is really interesting and can be further exploited.
Yeah, it sounds like it opens a lot of questions, like in a lot of avenues to test the question. So what are the next steps for this approach? So it sounds like you're tagging other clock genes. Do you anticipate ever using it in vivo? Is that a thing that could be possible, or what kind of next steps do you have?
People do in vivo. People have been done in vivo with fluorescence. Also, Mick Hastings in this meeting presented some engineered mice. I think with our cell lines, we have been the unique opportunity to have really first of all, it's a human it's a human cell line, and we have done other human cell lines, and I think we need to learn more about the human circadian clock and discover potential differences. It's a highly used model, and I think our expertise is more using these because in the cell biology we can also knock down things. We can use RNAi and stuff like this. So I think we have both approaches have pros and cons, and we are not planning to do mice.
So do you find that different cell lines have different properties when you are like quantifying the expression patterns of these clock genes?
Not so much in the human cell lines. So we have also done HCT 160 cells, which is a colon carcinoma cell, from which we knew that these cells also have a nice circadian clock. And we used that to kind of validate our findings in these U2S cells we have been using. And it's more or less the same. There are always slight differences, but the concepts are the same.
Cool, awesome. Well, it sounds like a really interesting approach. So switching gears a bit, I wanted to talk to you also today about the idea of chronomedicine. So this is something that was talked about a bit at the conference, and it seems like there's some interest in harnessing kind of the natural changes in physiology that occur during the day to target treatments for their optimal effect. And so it sounds like this has been a promising approach, but also that hasn't worked universally. I was interested to read your paper, developing a new tool to basically try to personalize chronomedicine. So basically, if I have this correctly, you built a tool to assess individual circadian time. Is that fair? Is that a fair characterization?
Yeah, that's a fair statement. I think the challenge is that we have two levels of complexity here. First is the circadian medicine per se, or chronomedicine per se. John Hoganish would say circadian medicine. This is that so many drugs have a time of day dependent efficacy and side effects, and so many interventions have a time of day dependent effect on side effects. It's wound healing, it's kind of cardiac operations and so on. Vaccination has a time effect and so on. So, however, so my motivation to go deeper into that, into the personalization of that, was although people have been looking for time effects, often they find time effects, but for most of the drug on the market, this is not, or this has been not translated into recommendations, and often also many drugs also fail in the development, really maybe relatively late when it comes from mouse to humans. And one of the reasons I proposed was that humans are so much more different from each other as a population than, of course, these inbred mice, because we all have different chronotypes. So some of us are early birds, others are night owls, many are something in between. So there is no one time fits it all. So even though you say you should take aspirin in the evening because of the COX-1 enzymes peaking at this certain time, what is evening for me? What is evening for you? It might be hours different. So we need to have a relatively easy and an easy tool to, before we treat or diagnose, to determine internal time. And internal time means in this case internal phase. So am I when my alarm clock rings in the morning at 7 a.m., is it for me morning or is it for me in the middle of the night? And I should not get up. So the principle is, how can we do that in an easy way? Usually people do so-called dim light melatonin onset. So the dark hormone melatonin rises in the evening, and this kind of prepares the body for sleep, and you can determine this exceeding this threshold of some concentration in saliva or blood, and people consider that as the gold standard. But it's really cumbersome, expensive, and you need to have the people in the evening in a dark room, and they have to give saliva every half an hour for many. So we need to have something which you can use one sample and that's it. And the idea was that since we knew that many genes are clock controlled, 10%, 20% of all the genes in any given tissue have a rhythm, and the phases are different. So we have genes which are morning, in the morning highly active, others are in the evening highly active, yet others are midday highly active, and others are at noon at midnight highly active. So if I take a sample of blood and find the morning genes high and the evening genes low, and the others in between, then I know this is an internal morning. But if I wake you up at 7 a.m. and your midnight genes are still high and your morning genes are in between and your noon genes are really low, then I know I woke you up in the middle of your biological night, even though your alarm clock says 7 a.m., but it's still night in you, so you should sleep. So that's the idea. And we have, other labs, 2 other labs have also gone this way, but I think the distinctive thing in our study was that we decided to use a special blood cell. So we just didn't take only these white blood cells, PBMCs as we call them. We sorted out the monocytes because we had animal experiments in the past when we were interested in immunology and the clock that showed us that these monocytes had an extremely high amplitude rhythm. And so our test was, I think, based on that, and maybe based on our multiplex RNA quantification technology. This together made it highly accurate. So we are different from this gold standard DILMO only by half an hour, but the DILMO itself also has a variability of half an hour. So we think we are pretty accurate. So you just give us a blood sample any time in the day, and I can tell you what time is it in you, and not on your wristwatch only, but in you.
So, so do you envision that this will be used to guide treatments, or do you envision like I would send you a blood sample so that I could get an answer of when I should go to bed and when I should wake up? Like, what are the applications that you envision for this approach?
Yeah, I mean, that of course there's different applications. One is chronotherapy, chronopharmacology. This is one thing. When should I get my drug? Second thing is helping sleep doctors to diagnose circadian rhythm disorders, and just whether somebody is really, because behaviorally often people kind of come to the sleep physician and say, and behaviorally they have a delayed sleep-wake phase syndrome, so they cannot sleep before 2 a.m. or so, and whether this is a clock dysfunction we can test with our blood test. But of course, this has also potential for a lifestyle product. If you just want to optimize yourself and get an idea when to do your sports, when to do your cognitive tasks, we have been developing, but it's unpublished, also a hair-based test, because it turned out that for a lifestyle product, this blood test is too logistically challenging, because in our study we kind of processed the blood relatively immediately, and then you cannot send it in like a 23andMe sample.
Yeah, that sounds really interesting. So one question I had is it possible to like entrain your internal clock? So if I do you think that I have an intrinsic clock that you can measure, or do you think that if I go to bed every day at 11 and wake up at a certain time, do you think that eventually I can shift my clock one way or the other? Have you looked at that at all with this tool, or?
You can shift your clock when you shift the light dark cycle. If you travel to Europe, then you have a different light dark cycle, and then you can shift your clock. You can also shift your clock when you change your light levels. There's this famous camping study of Ken Wright, where people, when they went out camping in and had kind of outer light all day, their internal clock became earlier 1 or 2 hours. And I've been starting with this test a study where we follow every week individuals for over the whole year. And I can already say, so I do it also with myself, and I can see over from winter to summer now, I've gotten 1.5 hours earlier because of the earlier sunrise. And we are really, it's an N of 2 study so far, and this is only among us, the preliminary results, but let's see how it develops. But it opens interesting opportunities. We could also really have generate data on students in school, because we are always advertising for the adolescents 16 to 20 years old. This is the latest time in their life, and they shouldn't school shouldn't start at 8 a.m. Yeah. So we could measure their internal clock at 8 a.m. and compare that with, for example, cognitive tests. Also not done yet, but it's an idea.
I mean, it sounds really interesting, exciting. I'm especially fascinated by the longitudinal profiling and how it changes with the seasons.
Yeah.
So, I mean, so my last question is, we've kind of touched on it a little bit, but just wondering what kind of other future directions you have going, like what other next steps, projects you have in the lab, or what do you think are the big open questions in the field to kind of conclude our chat today? But we've also,
yeah, I think we the lab will be kind of divided between these two interests. One is the even better understand the molecular mechanism of the clock, the cell biology. We want to learn more about really how the cogwheels interact, and we have been also a fascinating study in press where it's about coupling between different clock cells. And on the other hand, I really think the field and moves forward into circadian medicine. So I have now at Charité is a medical school, and I have many colleagues who are highly interested in joining the field and applying these insights in the clinic. So, for example, my colleague Claudia Spies has an ICU, and she designed new rooms where they had big light screens to make for these ICU patients a nice light dark cycle, and then the parenteral feeding needs to be adapted to kind of give people rhythms back, because in the ICU rhythms often get lost. And this is correlated with longer stay in the hospital, with a higher likelihood to develop a delire and so on. So that's I want to kind of push these in my institution and beyond, that we kind of use our knowledge to really translate it into the to the patient.
Well, it sounds like there's a lot of really important directions for this work. So I have actually one last question, which is kind of a fun one. So you said that you have been profiling your own internal clock. So have you changed your lifestyle behaviors at all? Have you changed your bedtime or anything in terms of, or like when you go to work?
No, I mean, it is difficult to always, if you measure something, you change something. This is the saying, right? So I'm not sure whether. So I don't change anything on purpose, but by kind of looking at the data, maybe I do something different. So I'm a little bit this year, for some reason, I'm a little bit earlier, but I'm also getting older, and you know, the older you get, the earlier you get. So maybe it's age kicking in suddenly.
Okay, well, it was really nice to chat with you today. So thank you very much. And I think we can wrap the interview.
Thanks, Luke.
So, yeah,
it was a pleasure.